everyone. Thank you so much for being here. Welcome to the Arthritis National Research Foundation's Researcher Spotlight webinar series. I'm Jenny Dunn. I'm the Senior Director of Marketing at ANRF, and on behalf of our Board of Directors, Scientific Advisory Board, and staff, I'd like to welcome you to our Spotlight on Scleroderma. We're so excited to have you joining us today. We'd also like to recognize and thank our corporate partners for supporting this series. Thank you so much to AbbVie for being a gold level sponsor and for your continued partnership and dedication. It's because of these partnerships that we're able to continue to pave the way for new innovations and learnings from our researchers, so thank you. Before we get started, we have just a couple of logistical pieces of information we want to share. This webinar is being recorded and we'll have it available on our website and YouTube channel in the coming weeks. And if you have any questions, please type them into the Q&A box. If we aren't able to get to yours, you can always email us at info@curearthritis.org after the webinar, and we'll make sure you get an answer. I am now honored to turn the mic over to our patient moderator, Amy Gietzen, who's here to share her journey with scleroderma. Amy?
Thanks, Jenny. Thanks, everybody. So really quickly, my journey with scleroderma began 23 years ago. I was only 19, and I cannot tell you how shocked and just confused I was during that time. But really, in the impact of it, I felt nothing. My emotions and my anxiety over it, it kind of just left me feeling numb. My first instinct was to pretend that it never happened. I never went to the doctor that day and never had x-rays, and I most certainly never sat down and was told I had a rare chronic illness with no known cause or cure. Nope, it did not happen to me. So I kind of went on with normal days of life because of that, and it was just easier than doing the hard thing, which was to face reality that this disease would most definitely be a huge part of my life. As far as treatment went, my local specialists, they really knew next to nothing about scleroderma back then, and the few that did wanted to treat me symptomatically and not really stop progression, which was what I was looking for. So I learned pretty quickly that my care would be subpar if I didn't start to take the reins and make a plan and put that plan into action. The first thing I did was I had to accept that this disease wasn't going anywhere, right? There was no magic pill to make it better. And second, I needed to find out everything and anything I could about what exactly scleroderma was and how it was affecting my body. And then I needed to find some doctors to help me, some experienced and innovative specialists that would try their best to keep me alive for as long as possible. So I pushed my feelings aside, concentrated at the task at hand, which was finding a scleroderma center as close to me as I could. And that took me about 5 years. Finally, when I did, it really changed my life. Gone was the girl who refused to acknowledge her disease. Gone was the girl who accepted mediocre care and pushed aside any form of support. And gone was the old me, for better or for worse. Finding that center and the scleroderma doctors within it literally saved my life, not just physically, but emotionally and spiritually. It forced me to come face to face with this disease and own up to the fact that I would have this for my entire life. It was not just going to disappear or quietly move on to someone else. And more than likely, it would be something that eventually took my life. The center opened my eyes to the world of the chronically ill. I started to take a more active role in my healthcare, and doing that, I eventually started to take a more active role in outreach and finding people with scleroderma who needed support. I connected with a lot of people online at first, and I felt like if I could help people from being affected less like I was, that maybe I could do some good. I started to work with the National Scleroderma Foundation, who eventually invited me to speak on Capitol Hill during Rare Disease Day. And after that, my awareness journey took flight. Over the years, I've spoken on panels for NORD in Texas, met with second-year medical students at Michigan State University. I was a keynote speaker for an interprofessional event in Albany, attended countless Capitol Hills in DC, And I established a virtual young adult space for scleroderma patients in their preteen and early adult ages. I've created panel after panel, and I've also spoken on FDA panels. I've taken so much joy in the work I've done. It's taken me 23 years to get to a place of ease, a place where I know I'm finally accepting this disease and I'm embracing all the good and the bad times I've had so far. Was it easy? Absolutely not. Was it fair? Not at all. Was I angry, hurt, devastated? Tick, tick, tick. Yes, yes, yes. It was and still is a hard journey to live a life with a disease like scleroderma. However, through the ups and downs of this disease, I've met so many amazing, incredibly strong people who do the work, who are patients, and who help us as patients, like some of these amazing doctors that you're going to hear soon. I've traveled all over the country doing the thing that I love, which is educating. And, you know, I've learned that I can't dwell on the past. 23 years ago, scleroderma was a word I could not even pronounce. Now it's a word I say with pride because it has allowed me to create memories, moments, and friendships that I will definitely cherish forever. Thank you, guys. So now I kind of want to move in and introduce our first speaker. So our first speaker is Dr. Mark Lee. He is a principal investigator at Yale School of Medicine, where he leads a human immunology research laboratory. He's also a board-certified transfusion medicine physician. In his clinical role, he helps oversee apheresis procedures as well as complicated blood transfusion needs. The Arthritis National Research Foundation is helping Dr. Lee work towards the major goals of his research, which include developing novel technologies to identify T-cell epitopes and identifying epitopes targeted by tumor-infiltrating T cells to develop novel T-cell receptor-based immunotherapies. So everyone, please welcome Dr. Lee.
Thank you so much, Amy. Thanks so much for sharing your story. It's really inspirational and motivational for me, and I imagine for other researchers out there. So I'll provide just a little more background about my training. So I'm a physician scientist by training, which means that after medical school and graduate school, I completed my residency in clinical pathology and then a fellowship in transfusion medicine and then additional postdoctoral research training. Before starting my own laboratory at Yale, where I study human immunology. And then, as Amy mentioned, I also am a physician in the Department of Laboratory Medicine, where I work in the blood bank at Yale New Haven Hospital, as well as work on the apheresis and cell therapy units in the hospital. So while I don't directly treat patients with scleroderma as part of my clinical work, through the research with ANRF, I collaborate really closely with Dr. Monique Hinchcliffe, who is a rheumatologist at Yale. She's a physician scientist herself, and she's the director of Yale's scleroderma program, and so really has the deep clinical expertise in scleroderma. And I believe she's been a participant on the ANRF webinar series that you could find on one of the previous videos on the website. So Amy provided, an amazing description about her experience. I'll just briefly review some of the background and causes of scleroderma. So scleroderma is a chronic autoimmune disease that's characterized by the excess deposition of collagen or scar-like tissue, often in the skin, sometimes in other parts of the body, that leads to progressive disability. So there are various classifications of scleroderma, including localized, which where the symptoms are more localized to the skin, and systemic scleroderma, which is also called systemic sclerosis. So in systemic scleroderma, the deposition of scar-like tissue in various internal organs, for example, if the lungs are affected or the heart, kidneys, or digestive system, this can lead to additional symptoms and in some cases can lead to premature death. So scleroderma, in terms of the overall impact, affects about 300,000 Americans. And the research need is high, as scleroderma has no known cure and can progress despite current treatments. So in terms of the symptoms of scleroderma, this includes thickened patches of skin that could occur on the arms, legs, face, or other parts of the body. The loss of joint motility, for example, in the hands with concomitant muscle pain or weakness. And if the internal organs are affected, then this could lead to symptoms such as shortness of breath or chest pain or difficulty swallowing or high blood pressure, among other symptoms. And the treatments of scleroderma often involve the use of broad immunosuppressive medications. This could include topical corticosteroids or corticosteroids given by mouth or intravenously. Sometimes other broad immunosuppressive medications are used, such as methotrexate or mycophenolate mofetil. And other drugs that are sometimes used include non-steroidal anti-inflammatories, vasodilators, and antifibrotic medications. So currently in clinical trials, there has been some really intriguing data on a new form of immunotherapy that involves depleting B cells in the body, either through something called CAR T cell therapy or an anti-CD3, CD19 bispecific. This has shown promising results, but in a small number of patients, and the data is still being collected there. So as maybe Amy mentioned, the exact cause of scleroderma, unfortunately, is still not known. However, many researchers have been studying the disease, and the current disease model suggests that in scleroderma patients, the immune system attacks the body's blood vessels, which damages them and causes inflammation. So normally our immune system is there to recognize and fight off infectious diseases. For example, if we get the flu, then the immune system recognizes that the flu virus is in our body, and then it mounts an immune response, including using cells such as B cells and T cells to fight the flu and get rid of the flu virus from our body. But in autoimmune diseases such as scleroderma, the immune system seems to attack itself for unknown reason. And so in response to that damage of blood vessels, fibroblasts get activated. They overdeposit this scar-like tissue, and that leads to the tissue becoming thickened and tight, and it could cause certain organs to not function like they normally do. So while the exact cause of scleroderma is unknown, there have been some major clues that researchers have found. One of them is human genetic studies, which have looked at inheritance patterns among patients with scleroderma. So in some of these genetic studies, essentially what they did is they took a large number of patients with scleroderma and they sequenced parts of the DNA in the genomes of the patients, and they took an equivalent number of healthy individuals and they took their DNA and sequenced them. And the questions the researchers asked is, are there differences in DNA sequences between patients and healthy controls? And in those types of genetic studies, intriguingly, they found a number of differences, including differences in genes that affect the function of T cells. But most prominently, they found a difference in a region of the genome called the HLA region. So I think this is fascinating because if you think about the function of the HLA genes, essentially what they do, if you look at the diagram on the right, is they present small pieces of proteins, which are called peptides, to T cells through what's called the T cell receptor. And this leads to the T cells essentially turning on. So this is believed to be one of the on signals of T cells in the body. So for example, when we get a flu infection, then the HLA proteins are supposed to present flu peptides that turn on flu-specific T cells. And then after the T cells get turned on, then they go off and they try to eliminate the flu virus from the body. What this means in scleroderma is not entirely clear, but one of the leading hypotheses is that the HLA genes in scleroderma patients actually present a peptide or protein that's part of our normal human body. And this leads to the activation of certain scleroderma disease-causing T cells. So those T cells then turn on and then cause tissue damage. So there has been a lot of interest in trying to figure out what are the components of this on signal? What is the HLA gene? What is the human peptide? And what are the scleroderma T cells that are involved in this on signal? And so the reason that there's a lot of interest from the research standpoint is that strategies that if we could understand this complex, then strategies to block this on signal may hold promise as a novel treatment strategy for scleroderma. However, for many years, people have been trying to answer this question, and it has been unfortunately unresolved. And so in collaboration with Dr. Hinchcliffe through funding from ANRF, we're trying to revisit this very challenging question. And the reason that we think our approach is different and may hold promise for solving this problem is that we're coming at it with new experimental techniques that we developed. In kind of broad overview, essentially what we're doing is we are studying tissue directly from scleroderma patients that has either been collected for clinical purposes or are small pieces of skin tissue that have been donated by patients for research purposes. And so what we're doing is we're taking those little pieces of tissue, we're looking at them under the microscope, and we're looking for this type of infiltration of immune cells, including T cells, in that tissue, particularly T cells that have infiltrated around blood vessels, since that is one of the driving theories of how scleroderma initiates. And then using specialized techniques in the lab, essentially what we could do is we could characterize the T cells in the tissue samples, and we could recreate the T cells in our lab, essentially grow the T cells in a sterile dish. And by doing that, essentially that gives us the ability to test hundreds of thousands of human peptides for the potential to turn on the T cells that we created in the laboratory. Ultimately, with the goal to identify this type of HLA peptide T cell receptor complex that leads to T cell activation in patients. And so going back to our future goal, if we're successful to solve this problem, what we would be interested in is developing ways to then turn off those T cells, either using approaches called regulatory T cell therapies or direct ways to eliminate pathogenic T cells from the body. And so this work has been led by a number of people in my lab, including Mimu Mosharov, Matila Kasby, and Berk Hayashi, with a really close collaboration with Dr. Hinchcliffe and her team, particularly Eliza Tsou, and really couldn't be done without the support that we received from the Arthritis National Research Foundation. So we're really grateful for their support. Thank you.
Thank you, Dr. Lee. That was great. Fantastic. Super, super intriguing, and really, it was really making me have lots of questions towards the end. So if you are like me and have a ton of questions to ask Dr. Lee, please put the questions in the Q&A box on the bottom of your screen, and we'll get to them at the end of the presentation. Now I'd like to introduce Dr. Tsou, Dr. Eliza Tsou. She's an assistant professor at the University of Michigan. where she focuses on researching scleroderma. Her work aims to understand the vascular problems that occur early in the disease, which leads to blood vessel damage and other complications. With a background in both pharmacology and rheumatology, she collaborates with other experts to study autoimmune diseases. Dr. Tsou has published over 60 research papers and has been funded by major organizations, including the Department of Defense and the National Institute of Health, as well as receiving support from ANRF. Please welcome Dr. Tsou.
Thank you, Amy. Good afternoon, everyone. It is my great pleasure to be here to share our research with you. I would first like to thank ANRF for the invitation. I received funding from ANRF way back when I was a postdoctoral fellow, and the work that I'm presenting today is actually based off the work that was funded at the time. So for that, I'm really grateful. I am a PhD scientist. I spend my everyday life in the lab researching scleroderma. And today I thought I would share with you a cute little story that we developed over the years, focusing on vascular aging in this disease and what we have learned from before and what we know now. So let me try to advance. As Dr. Lee has already alluded, and as all of the patients who are calling in, living day-to-day life with this disease, we know that systemic sclerosis or scleroderma is a rare multi-system autoimmune disease. In Greek, sclero means thicken, derma means skin. So on the outside, these patients do have a lot of skin involvement. However, we do know that this disease is more than skin deep. This tissue thickening process, which we call fibrosis, can actually be spread into many internal organs. And it's these internal organ involvements shown here in this picture in the cartoon on the right that causes a lot of the symptoms that the patients are suffering every day. Treatment is fairly limited at this point. It's mainly just treating the symptoms, but not the underlying cause. And so as scleroderma researchers, we're interested in trying to understand what is happening inside the body of a patient. And there are three major components in this disease, including immune system activation, blood vessel damage, and tissue thickening or fibrosis. So if you look at the blood vessels on the bottom of the screen, this blood vessel is usually the first place where the tissue injury happens. And the blood vessels is made up of blood vessel cells or vascular cells. And these blood vessel cells here form these leaky blood vessels that can allow these activated immune cells to escape from the circulation and enter the tissue. Once in the tissue, they can produce a lot of these pro-inflammatory mediators and to make this tissue very inflamed. In this environment, tissue thickening cells, which are on the top of the screen, they become activated and they can become these red hot myofibroblasts that are super activated. They can produce a lot of these tissue hardening substances, including collagen, that can lead to tissue fibrosis. In my lab, we're interested in vascular changes in this disease. And as patients, you probably suffered from one or more than one of these vascular manifestations. One of the most prominent one is probably a Raynaud's phenomenon on the left. This is usually one of the first sign of symptoms that patients suffer from: changes of color of your fingertip because of changes of blood flow, maybe because it got really cold today. And at some point in the clinic, the physicians probably looked at your nail fold capillary, and you went through something called the nail fold video capillaroscopy to look at the base of your fingertip, just a fingernail, just to see the malformed capillary beds in the fingernail. And that's a direct indication that there is indeed vascular changes in these patients. Other vascular manifestations include very painful digital ulcers, pulmonary arterial hypertension, as well as scleroderma renal crisis. And so if you're interested to know what type of vascular changes are happening in the patient, and this slide summarizes the two types of morphological features of vascular involvements. One type is called the fibroproliferative vasculopathy, and on the right side of the screen is destructive vasculopathy. On the left side of the screen in this figure, it just shows you these normal physiology of different blood vessels. So they come in different sizes, but they usually form these very tight seals so that nothing bad can leak out of the blood vessel. For the proliferative vasculopathy, this usually happens in small arteries, and you can see there usually is a lumen in this artery and it allows the cells in the blood to flow through. However, in the histology slide, you can see that this small artery and from this patient, the lumen completely disappeared and it's being replaced by these overgrowing cells from the blood vessel wall. So usually fibroproliferative vasculopathy causes pulmonary arterial hypertension. On the right side, there is destructive vasculopathy, which is the complete opposite on the left. So these usually happen in capillaries. Capillary are smaller blood vessels, usually have one layer of cells, but they're tightly sealed so that nothing can leak out. However, in scleroderma patients, we do see that these capillaries look like thinner. Sometimes they have gaps in between cells, and this is when the immune cells can escape and enter the tissue. And so for scleroderma research, I would say we're lucky in a way that there is a lot of skin involvement in this disease, and skin biopsies are relatively easier to obtain compared to a lung biopsy or a kidney biopsy. So in our lab, we obtain a lot of these skin biopsies, and I'm sure if the patients on call probably donate a skin biopsy either for pathology or for research. And the size of the skin biopsy that we collect is usually very small. They're usually like the size of an eraser on a pencil, 3 millimeter in size. And from this tiny skin biopsies, we can learn so much about this disease. And in the middle of the slide shows a cross section of the skin. You can see the upper layer is called the epidermal layer, is where it faces the outside world. And so if you get tan, it's tan in this layer. And the dermal layer is follows in the middle, and this is where all the good stuff is. You have the blood vessel that flows through, you have your hair follicle, you also have your sweat gland, and then the bottom part is the fat tissue. From different part of the skin, we can isolate different type of cells. So for example, from the epidermal layer, we can isolate what we call keratinocytes. They're more bigger cells and they're more masculine looking. From the dermal layer, we can isolate tissue thickening cells or fibroblasts. And you can see under the microscope, they look more elongated. And with the staining on the right, you can see they're red hot, very active phenotype that kind of make them a little bit evil looking. From the blood vessels, we can also isolate vascular cells or endothelial cells. And under the microscope, you can appreciate that they look very different from the fibroblasts on the top, and they look more rounded and more cobblestone-like structure. And so since we're working with the blood vessels, I want to spend a little bit more time about these vascular cells. And on the right, you can appreciate that this is one vascular cell line isolated from a scleroderma patient in a petri dish, and they do grow over time. You can see they sometimes move around in the dish. These cells in our lab, we call them princess cells because they need a lot of attention and need to be pampered all the time. And we can use these cells in a lot of different experiments. And this is where I think my lab has a unique position to do a lot of these research because we can grow them in a petri dish and work with them. And so once we have these cells in hand, we started to characterize them and see what kind of behavior that they have when they're grown in a petri dish. Do they show the same phenotype that what they're doing in the patient body? And so one of the first things that when we think about vascular cells is that in the body, they form blood vessels. And so in a petri dish, we can do something very similar. This is called a matrigel tube formation assay, where we plate these cells on a gel, and over time, they would start forming tubes. So you can see on the left side in this movie, this is from a healthy subject, and the right side, this is vascular cell from a scleroderma patient. They're starting to form tube-like structures, just like how they're forming blood vessels in the body. You can appreciate that the patient cell line is trying to make tubes, but sometimes the tubes don't really form as well as established as what the healthy control looks like. And also, if you look at the nose where the tubes are stretching out, in the scleroderma patients, it looks smaller, skinnier, not as well structured as what we see in a healthy subject on the left. So from this, we know that the scleroderma vascular cells are unable to form healthy tube-like structures in a culture dish, similar to what they behave in a patient's body, that they don't form very healthy blood vessels. Another feature of these vascular cells that we work with is that they form leaky monolayers in a culture dish. So what we did is we cultured these cells in a dish. The top is a healthy subject and the bottom is a scleroderma patient. We stained a protein called V cadherin in purple, and this protein kind of labels the cell edges. So the blue dot is a nucleus. You can kind of see on top that the cells, the purple kind of lines up where the cell edges are, and it kind of separates out the cell from one cell to a neighboring cell very well. However, in a scleroderma patient, you can see that there is some purple in there, but the edges are not as prominent. Sometimes there's disappearance of this purple edge. And what this translates into is that when we do a permeability assay where we plate the cells on the bottom of the trans well, and then we add samples on top to see how much can run through the monolayer of cells to the bottom of the well, we see that in scleroderma patients, there are more substances that can see through the monolayer of these cells that they form. And similar to what we see in capillaries of patients, that they form these leaky blood vessels that allow things to see through more. Another very unique feature of these vascular cells is that they undergo something called endo-MT. It's a very fancy name, stands for endothelial to mesenchymal transition. What this means is that these vascular cells have a serious identity issue. They don't want to be vascular cells anymore. They kind of want to be more like tissue thickening cells. So if you look on the left side of this figure, these vascular cells usually have their rounded appearance. However, when they're going through endo-MT, they acquire more of the spindle-like, longer appearance of a fibroblast. They lose their vascular markers and then gain all these fibrotic markers. So on the right side of the screen, you can see this is one example. We have cells isolated from a healthy subject on the left, and on the right is a scleroderma patient. The blue dot is a nucleus. The green on the left is a vascular marker. So on the left side for this healthy subject, you can tell that these are happy vascular cells with a lot of vascular marker staining. On the right side, you can see that there's not that much green anymore and more of the red color. The red color is a staining for a skin thickening marker. So these are vascular cells, but they're kind of gaining more of a tissue thickening phenotype. And this is very unique in vascular cells from a scleroderma patient. So what is causing all these abnormal properties and phenotypes that these cells are showing? And so here I want to introduce you to a concept called cellular senescence. Senescence, sene in Latin means old age, aging, older. And so senescent cells means that these cells are older cells. You can think of these cells as zombie cells if it's easier to understand. These zombie cells are malfunctioning cells that they pretty much don't grow, but they don't die. So over time, there's these large number of zombie cells that can build up in your body through aging. So the 45-year-old me has more zombie cells than the 35-year-old me. This is a normal process. However, in scleroderma patients, there are just more of these zombie cells in the vasculature. And if you look at the cartoon in the middle, the zombie vascular cells can spit out a lot of these SASP proteins, and these are pro-inflammatory proteins that can spread their zombiness and infecting the neighboring cells to make the neighboring cells zombie cells. They also have an ability to self-reinforce their zombiness, so they stay as zombie cells for a very long time. So if you have a son like mine who is very into Minecraft, which is a video game, I was educated that there are different types of zombies in Minecraft. There's this real zombie, a pig zombie, a villager zombie. There are different identities. In the field of senescence research or zombie cell research, it is a little bit messy to kind of how to define a zombie cell as a zombie cell. So there is a lot of discussion about how we can identify and make sure that we validate that what we're seeing are really zombie cells. So in our lab, we spend a lot of time trying to validate our findings, make sure that we look through a lot of these zombie or senescence markers to make sure that indeed the cells that we isolate from patients are indeed zombie vascular cells. And this is not just happening in a petri dish. So if we look at scleroderma skin, from a skin biopsy, we can get cross sections of the skin and do different stainings. And in this particular slide, on the top is a skin section from a healthy subject. On the bottom is from a scleroderma patient. The green color is a vascular marker. So you can see that that's a blood vessel right there in the top left. And on the bottom is the same. For the red color, we're looking at zombie markers. You can see that there's prominent overlapping of the green and the red in the scleroderma patients in the bottom right picture, but the red is essentially not there in the healthy subject. And so this indicates evidence that these vascular zombie cells are indeed observed in scleroderma skin. So what we think is happening in scleroderma is that in our body, there are different type of vascular cells. We start on the left. And then for some reason, there's this trigger that induces cellular senescence in these patients. And that leads to expansion of these zombie vascular cells. These zombie vascular cells can lead to vascular changes. Remember the proliferative and destructive phenotype that we talked about. Through spitting out SASP, these very pro-inflammatory proteins, and these cells can undergo this identity crisis, endo-MT, to become more of a skin thickening cells, and that leads to tissue thickening on the bottom of the figure. And so these zombie vascular cells can contribute to the vascular changes and also tissue thickening. Both of these lead to the clinical manifestations that we see in these patients. And so in our lab right now is actively trying to figure out what is that trigger that is increasing the expansion of these vascular zombie cells. And also to figure out whether there is a way that we can target these vascular zombie cells to get rid of them so that we can relieve the patients from suffering from a lot of these vascular manifestations that they're dealing with every day. So with that, I would like to thank the patients who kindly donated their skin biopsy for our research. You know, none of this would be possible without the cells that we isolate from the skin biopsies from patients. So I'm really grateful for that. At the University of Michigan, I co-direct the Scleroderma biorepository, and I want to thank the staff there for isolating the cells and maintaining them. The lab members and colleagues and collaborators from the University of Michigan. And last but not least, my funding sources, especially from ANRF, who believed in me very early on in my postdoctoral time. And I don't think I would be here today without the funding that they provided me back then. So I'm really grateful. Thank you. I'd like to take any questions if you have.
Thank you, Dr. Tsou. I can really appreciate you comparing us to zombies, because sometimes I do absolutely feel like that. So now it's the Q&A kind of portion of this webinar. So like I said, you can put your questions in the Q&A box on the bottom. We have our first question, which is what factors influence the progression of scleroderma? And are there any early indicators that suggest a more aggressive form of the disease? So either of you could jump in, Dr. Lee or Dr. Tsou.
I can chime in. I think as Dr. Lee alluded in his talk, there's definitely genetic component in this disease. I think there is what my understanding is what influences the progression is you need to have the genetic background. And then there also have some environmental triggers that it's called epigenetics. So it's like above of your genetic backbone, and together with the predisposition, with the epigenetic and environmental triggers that kind of starts this disease. And maybe that plays a key role in how the disease progresses. I don't know if Dr. Lee wants to add on.
Yeah, no, I was thinking exactly the same thing, that probably there is a genetic component that patients are inheriting different genetic variants that may ultimately influence the way the disease progresses. I think things like response to treatment, and kind of like even what Amy was saying in terms of her own experience, finding like a really good care center probably makes a difference in terms of outcome.
Sure. Yeah, it did for me, so I'm sure it could for a lot of other patients. Great. The next question is, what scleroderma discoveries are you most excited by and that you think patients like myself and others would or should want to know about? I mean, the CAR T cell seems amazing.
Yeah, I think the CAR T cell, even though it's related, but not exactly what we do. I think like when I read about that, and I think it's still early days, and it's not there's definitely some pros and cons of the therapy. Like we all need B cells, so depleting B cells has its own dangers. But I think the remarkable progress that a small number of patients did experience was different than we've seen for other therapies that have been tried. And I think that there has been a lot of interest in how that field will develop. I think I'm personally interested in it because um, as I mentioned in terms of the treatments, a lot of them are broad immunosuppressives, and broad immunosuppressives themselves have a lot of bad side effects. And I think one of the goals in human immunology and in studying autoimmune disease is how do you more specifically just block the one part of the immune system that is doing something wrong, such that you're not more susceptible to other infections or other things that the immune system normally does, but you only block the bad part. I think that really is the goal and the future of immunotherapy research, but it's a very challenging one.
Sure. Thank you, Dr. Lee. Dr. So, do you feel like there's anything new and exciting coming down the pipeline on your end that would
Well,
perk?
I can tell you that the CAR T therapy is one of the hottest thing right now. University of Michigan actually is starting several clinical trials, specifically looking at CAR T therapy in systemic sclerosis patients. It's really in its early stage. You know, we have some reports from Europe that CAR T therapy is really effective in patients with lupus, and they have a small group of scleroderma patients that seem to be very promising. So I think this is really, it really excites me that, you know, it does seem very promising, although it's in its early stage. And Dr. Lee did say that there's, you know, with cellular therapy, there's always some risk. And so definitely the patients, if you're interested in, you can read more about it. And I think this would be a revolutionized therapy for us to look forward to.
Yeah, exactly. Thank you. Okay. Another question we have is, how do the abnormal T cells or zombie cells cause the pain that we as patients deal with?
I think the pain, I'm not a clinician, so maybe Dr. Lee can help me, but I think the pain comes from the tissue thickening, the fibrosis process, right? And so at least for T cells and the vascular cells that we see, it happens really early on in disease that feeds into this fibrotic phenotype that happens more towards the later part of the disease. So the research that we do, that at least for the vascular part, I'm trying to figure out whether in an early stage in the disease that we can stop this from progressing into the fibrotic stage. And for T cells, Dr. Lee can chime in, that it's just trying to figure out that initial step, right? And see if we can stop it from progressing to some degree of fibrosis.
Sure.
Yeah, I think that's a tough question to ask
Yeah.
to answer scientifically in terms of the progression from some of the initiating events that
Right, because I think
I talked about.
all patients with scleroderma are so individualized that some people may have more pain
Individualized.
than other people do. So it's hard to pinpoint.
Mhm.
And your pain could be coming from digital ulcers, it could be coming
Visual
from
ulcers,
stiffness in your
contraction.
joints, and so yeah, I
Exactly.
agree with that. Let's see. Okay. If someone has multiple autoimmune diseases, could treatments for those like infusions make scleroderma more active or get worse? Um, I guess so. Would you have to rob Peter to pay Paul in terms of like treating scleroderma instead of this other multiple autoimmune diseases, I guess is what they're asking, or
Yeah, I think that's also a really complicated question. I think that does point to, an interesting observation that many autoimmune diseases, um, they share some of the same risk variants, and so some patients can present with multiple autoimmune diseases, really pointing to, a possible component of genetics. In terms of the treatments, as I talked about, some of the treatments are broadly immunosuppressive.
Right.
Um, but I think in terms of the specific medications, those are really decisions that an expert clinician would be thinking about in terms of interactions and get very challenging clinically. But that's I would defer to the treating provider there.
Yeah.
Yeah, I think it's so great to have like a team of people that work on your case and continually speak to one another so that these type of questions can be answered right away and you can kind of get on board with some type of treatment. Um, let's see. What inspired both of you to do research in this specific area of autoimmune and more specifically scleroderma?
Yeah, for me, I think it started as a graduate student. So I took some graduate courses in immunology. I thought they were super fascinating about just the complexity of the immune system, how it fights off infection, and for my PhD work, I was specifically looking into the role of DNA and genetic variants in developing autoimmune diseases. And then when I was doing my postdoctoral research, I worked to develop an experimental approach that I talked a little bit about during my talk that I thought actually we could go back and study the genetics using this new approach that we developed. And so then when I started my own lab, I reached out to Dr. Hinchcliffe. We had a really great discussion about trying to unlock some of the mysteries of scleroderma using these approaches. And I think one of the things that inspired me is Dr. Hinchcliffe herself, since she's so passionate about scleroderma care and advancing treatments. And I found that really inspiring. And so we started working together, and that's kind of how I've gotten to where I've gotten.
That's fantastic, Dr. Lee. Thank you. Dr. So,
Um,
what
so
made you pick scleroderma?
and so I come from a drug background, so my PhD is actually in pharmaceutical sciences. So when I got my PhD, I was really looking into like drug treatment. However, when I was looking for a postdoctoral fellowship, I stumbled upon scleroderma. To be honest, before that, I've never heard of this disease before. I have to Google it before my job interview, but I just see it as a disease that needs so much, like especially the vascular part. It just needs, there's so many things that we don't know. And I was lucky enough that during this postdoctoral fellowship at University of Michigan, I was able to interact with patients and really understand, through the conversations, although I'm a lab person, but I can always kind of connect with patients and know day-to-day life, what they're struggling from, and what are the things that need to be done. And that really motivated me to stay in this field. It's since, 10, 15 years. So I'm still here. I'm still trying to work through this disease and try to understand what this disease is about.
That's amazing. Well, we're both really, all of us are really lucky to have both of you. Just the facets you guys go to kind of dig into the research and find new and exciting things for patients to be excited about is really great. So that's amazing. Okay, let me see here. Should scleroderma patients have a vascular specialist as part of their standard care? That would probably be a Dr. So question.
um, ideally, yes, but I think a lot of these vascular complications can be handled by the rheumatologist that you see. a lot of these, for example, pulmonary arterial hypertension, lung involvement, as you should have a pulmonologist that can help you with that. So ideally, yes, but I think our rheumatologists are doing a great job knowing what drugs to prescribe to help with your digital ulcers, with your Raynaud's. And so I would say stick with your rheumatologist. They know what they're doing and they take good care of you.
Perfect. Okay. With mild interstitial fibrosis, is it safe to take an immunosuppressant medication? Maybe Dr. Lee, you want to take that one?
That's a question I probably would defer to
Yeah, I'm not a clinician, so I can't
Yeah.
really give any medical advice. And so that would be a more specific question for your rheumatologist, maybe.
Perfect. Will therapies to block interferon and alter T cells affect scleroderma negatively?
That's a great question. That's a complicated question. So T cells play a really important role in our body, whether you're healthy or whether you have scleroderma, in fighting infection. And so we definitely, any therapy that will significantly alter all of the T cells in our body will definitely have negative effects, which they'll make people prone to infection. Specifically, the hypothesis that I think we're operating on is that there are, to put it simply, what we believe to be bad T cells that are involved in the disease process, and that the hypothesis is that if you eliminate them, that it will have a positive effect on scleroderma. I think the analogy that we could think about there is in a disease like type 1 diabetes, which is a different type of autoimmune disease. And there's more data where if you block certain T cells in type 1 diabetic patients, then they have a favorable response. But modulating the immune system in general, I agree, is very complicated, and this is something that ultimately, if new therapies are developed, would have to be tested in initially
Absolutely.
early phase 1 clinical trials to look at safety.
Yeah, absolutely. Thank you. Okay, so our final question is kind of a two-parter. What have been the biggest challenges in your research so far? And the second part is, can patients be a part of your research, and how would we go about doing that if you're interested? So I'll give it to you, Dr. Lee, first, and then we'll take to Dr. Tsou.
Yeah, I think one of the big challenges in scleroderma, as we all kind of mentioned, is that there's so much unknown, and so being, newer to the scleroderma research field, it's a huge challenge in and of itself to try to understand the disease. Yeah, something I talked with Dr. Hinchcliffe about, that this is so hard, but that on the flip side, because there's a lot unknown, hopefully discoveries will be made that drive progress in understanding. In terms of patients, yeah, definitely. So Dr. Hinchcliffe enrolls her patients all the time for various research projects, including the one that I'm working with her on. Patients have been a key part of what we do, and we are just eternally grateful for their contribution.
Thank you, Dr. Lee. Dr. Tsou, if people were interested in your research, how would they join up?
So I would say one of the biggest challenge right now, I think, is funding, right? We all need resources to run our research operation, and research can be so expensive and so costly. So this is where we're so grateful for ANRF to be part of our journey, where these foundation fundings really help jumpstart a lot of my for me, it jumpstarted my research program, and as a junior faculty where research resources are quite limited. So I think having ANRF on our side is great. It's great for the scientists. It's also great for the patients because they are advocating for the patients as well. Can patients be part of research? Absolutely, right? So I feel like scleroderma patients are the sweetest patients ever. They do anything to help us understand this disease, and we're really grateful for the time and the skin and the cells and the blood that they contribute to the research. So definitely there are a lot of different opportunities for patients to participate, not just for research, skin research or vascular research. There are a lot of studies that are looking at cognitive changes. We have a patient advocacy, I can't say that name, patient group that really at Michigan that we're looking at, whether a different stimulation with, video games or whatever activities that can boost the cognitive perception of these patients. So there are a lot of different opportunities. If you don't want to donate your skin biopsy, totally fine. There are other studies that you can participate that you feel like you're helping the research move forward.
Amazing. Thank you guys so much for all that. And I'll hand it back over to Jenny.
Thank you so much. First, I just want to thank our fantastic moderator, Amy. It was wonderful to hear your story. We're so grateful to have you here. Also, our incredible researchers sharing their work today towards understanding scleroderma. As we all know, research is the only way that new treatments can come to market and the only way we can get to a cure. So we so appreciate the work you're doing. You can see firsthand the impact ANRF has supporting these incredible individuals in their quest for a cure. I'd like to thank our partners once again, AbbVie, for their support of this webinar, and very much would like to thank everyone who joined us today for this event. If you enjoyed this webinar, we kindly ask you to consider making a donation today by visiting curearthritis.org/donate. Every contribution helps and gets us that much closer to a cure. We'll be sending our live attendees a survey after this event. Your feedback helps us find direction for future webinars, so please share your comments with us so we can continue providing important and relevant content for you. And please don't forget to sign up for our next Researcher Spotlight webinars. We'll be live at the same time on October 16th, looking at lupus, and October 30th, looking at osteoarthritis. Thank you so very much for coming. Again, thank you to our presenters, our moderators, and please remember to follow us on social media to stay connected. Have a great day.